welcome to another important class for my A levels biology lectures. And in today's class, what we people are gonna look that we would try to be understanding that what kind of association or what kind of interaction that is gonna exist between the multiple organelles that we have got, no matter whether that is an animal cell or a plant cell or a fungal cell. In that association, we have to be looking in terms of two of the organelles, not two, but rather than three of the organelles. Among them, we have the nucleus. After the nucleus, we be looking into the rough endoplasmic reticulums. And after the rough endoplasmic reticulums, we would have the third component coming here. And that third component that we earlier identified in the lecture of the ultrastructure of an animal cells. And this is what we were referring to this as the Golgi apparatus. Now, the situation that we have to consider is that, okay, fine, if we are convinced this with this fact that we are the living organisms, and as far as these living organisms are concerned, so these living organisms they have to survive. And the survival of the living organism is entirely based on that within their bodies there must happen multiple of the chemical reactions. And if these multiple chemical reactions they would not happen, then the living organisms cannot survive. Other than that, the second situation that we have to consider is that these living organisms they basically have to produce or they have to be developing the kind of the characteristics that lie on the outside of the living organisms like that we see with our own eyes. So they are termed as the phenotypic characteristics we say. Okay, so you see that we have to bring about a lot of chemical reactions and we have to be developing our outer observable characteristics or our phenotypes. And we will see that all of this actually is possible due to the presence of the nucleus we have, due to the presence of the rough endoplasmic reticulum we have, and the Golgi apparatus we have. Because for the survival of the living organisms, the protein making or the protein synthesis is important. If the protein synthesis is not taking place, then ultimately the living organism they cannot accomplish these, and if they cannot accomplish these, then we cannot be living at any cost. So we have to be thinking of a situation that how come the nucleus, rough endoplasmic reticulum, and the Golgi apparatus they are responsible for making up of the proteins, the enzymes, the outer feature that we have, the hair we have, the fingernails we have. So entirely like all the mechanisms, the complex mechanisms of the body, they have to be carried out. So here is the key to that. Now, what you need to remember is that if all of these scenarios that we have, like I have said at the moment, so if they are revolving around the making of the proteins, then we must know that how come these proteins they are made. So the situation I'm creating at the moment is that here within our cells, what we have got is this double membrane organelle. And this double membrane organelle that I'm making at the moment, you know it from the previous classes, that this would be entitled as the nucleus. Okay, and in close association with the nucleus, what we further know is that there would be the presence of this network of the membrane that we have, and these network of the membranes they had got on them some small spherical structures, and these small spherical structures we know that they are referred to as or they are entitled as the ribosomes. So these are the ribosomes, and overall the structure that we have got of these membrane and having the ribosomes on them, then collectively this would be referred to as a rough endoplasmic reticulum. Now, if I were to move further, then if you remember, I made another component for you and explained it to you in the previous classes, and that was an organelle, and this organelle it used to appear something like this. Like we would have some stalk of membrane-like structure, and the stalk of the membrane-like structure, you know, that this what I'm doing at the moment, we need to be giving this a name, and the name would be the Golgi body. So here we have got this Golgi body with us. Moving further, I would make some vesicles along with the Golgi body, and these vesicles, we gave them the name earlier. And the name of these vesicles, they were the Golgi vesicles, and the Golgi body that we have here, and the Golgi vesicles that we have here. So collectively, they are making the Golgi apparatus. Moving further, we know that these all the cellular structure they would be enclosed by this membrane, and the membrane that we have got over here. So this membrane, you must remember that this is entitled as the cell membrane, we say. So we have to create a situation where something must happen in between these organelles that would be making the proteins for our body, and by making of those proteins, we would be able to carry out multiple chemical reactions, and we would be making our outer observable features and even the internal organs. So now you just feel like that this nucleus that I have drawn for you, so this nucleus contains the heredity information in it or the genetic information in it, and this heredity information or the genetic information, this has come from the parent, and the genetic information simply lies in the form of the DNA. What you need to know is that this DNA has the instructions for specifying the sequence of the amino acids that we have to be making within the proteins. Okay, this is the same way. Like if I were to tell you an example that in my house I have got this one room, and then at some other part we have got this second room. Okay, by some mean or the other, there was a day that you had to stay in the kitchen and your mother had to stay here in this room. Now you were supposed to be making some dish, but you do not know that what kind of an ingredients you are required to be adding to be making your dish, and those you could say the ingredients, kind of the instructions that have to come from your mother. So this means that if we were able to be telling you, or if your mother was able to tell you the recipe for the dish, so if you would follow that recipe and if you keep on adding the ingredients, then you would end up making a dish out of it. So likewise, when I am talking about this nucleus, so this nucleus has a DNA, and in the DNA we have the instructions for what? For making the proteins. Okay, so those instructions they have to be carried to these small spherical structures that we have here. These are the ribosomes. So what actually happens is that from the DNA, the instructions that we have got for making of the protein, they would be copied on a molecule, and that molecule is entitled as, or this is called as a messenger RNA molecule. This is the same way. Like if I had to carry the instructions from the mother to you who you were standing in the kitchen, so there has to be some messenger here because mother herself cannot come to you. Likewise, the genetic information that we have in the nucleus, it itself cannot get out of the nucleus, so this has to be copying its information on this structure that we have here, and this structure is the messenger RNA. Now what happens next is that this messenger RNA will leave the nucleus through the nuclear pores, and after leaving through the nuclear pore, this would come and it would insert itself into the ribosomes. For the ribosomes, you just need to know is that they are responsible for making up of the protein. Now, how the protein is being formed in our body, that we would require these small units. These small units, they are the amino acids. And these amino acids, when they would link up with one another, so we would be making the chains out of them. And these chains, we call them as the polypeptide chains. And these polypeptide chains at the end, they would be converted into the polymers, and these polymers are the protein, and these are the protein that I'm required to make. Now, depending upon the dish, the list of the ingredient can change. So, depending upon whether I have to make my fingernail or whether I have to make my enzyme, so the sequence of those amino acids to be making the protein, they must change. So, we must know that what lies within this messenger RNA that would change the sequence of the amino acid, and every time we would be making a different protein. So the situation is that here every time when some specialized protein has to be made, so this messenger RNA brings some different instructions or it brings some different sequences from the DNA. And when the messenger RNA would come here, so what the ribosome is going to do that it would start to link the amino acids together. And when these amino acids they would start linking with one another, so they would lead toward the formation of the chains, and these chains they are termed as, or they are referred to as the polypeptide chains, we say. So what we must understand is that at the ribosomes that we have got, so here the protein that we have made, so this is a rough protein, like the function of that protein is yet not decided. Now I have to be modifying this protein. I need to be making this protein functional. So what I am supposed to do, that this rough protein that we have made onto the ribosomes, so we would pack it up into the vesicles, and these vesicles they would pinch off from the membrane of the rough endoplasmic reticulum. So this vesicle that I am making out here, so this is entitled as the RER vesicle, and the function of the RER vesicle is that any of the rough protein that we have made onto the ribosomes on the membrane of the rough endoplasmic reticulum. So these vesicles they would carry it toward this Golgi body. And as far as the role of the Golgi body is concerned, so what this is supposed to do, that this would modify the protein. This would fold the polypeptide chains differently. This would cut some amino acid out of the polypeptide chain. And as a result of that, a different protein would be formed. And that protein, depending upon if this is coming into our fingernail or onto the outside of the body, it would be making the skin. And if this would be retained within the cells, so it would make some enzymes for the cells. And this is how all of things would be accomplished. So just see that the RER vesicle that is coming from the rough endoplasmic reticulum, so this fuses with the Golgi body. And when this fuses with the Golgi body, so here the situation that we have to be generating is that here the rough protein has come where to these Golgi bodies. And these Golgi bodies, what they would do next, that they would modify this protein. And I have told you that that modification could either be differential folding of the protein, that could be cutting some amino acids out of it, or to this protein we can attach some other fat parts or we can attach some other protein parts. So a new kind of a functional protein would be formed everywhere. So when the protein goes into the Golgi body, so here this would be modified. Now, after the protein has been modified, now that protein would be packaged up into these Golgi vesicles, and these Golgi vesicles they would have the final protein in it, and that final protein, these Golgi vesicles, they carry it toward the cell membrane of the cell. And when these Golgi vesicles they have come to the cell membrane, so they would fuse here. And when they would fuse here, so as a result of that, the situation would be that the protein that we have got, this protein would be released out of the cells. And when this protein, when we are going to be releasing it out of the cell, so this could be either some enzyme, it could be making your body structure, it could be making the hemoglobin protein or any of the protein that we require for what to be carrying out multiple chemical reaction, or we were to be developing our outer features, even our nails and the skin and the hair and all of the stuff, even the muscles of our body. So whenever this has to be formed, so this mechanism has to follow. So if I were to summarize, if you have to be making a protein, then the instructions from the DNA they would be copied on toward the messenger RNA. The messenger RNA will leave the nucleus. This would go and would insert itself into the ribosomes. On the ribosomes, amino acids they would be linked by the peptide bond, and the linkage of these amino acids this would depend that what kind of the instruction this messenger RNA has brought. We would end up making the polypeptide chains. This is a rough protein. The rough protein, we will pack it up within the RER vesicles. These RER vesicles, they would go further and then they would fuse within the membrane of the Golgi body, and within the Golgi body, the protein would be modified. After we have modified the protein, then we would pack it up in the Golgi vesicles, and these Golgi vesicles will then move toward the cell membrane. They would fuse here, and the final protein would be released out. So this is what is a basic mechanism or an association between the nucleus, between the RER, between the Golgi body and the Golgi vesicles. And this is the right process on which our life actually depends on. I hope you would be clear with it. So see you in the next class.